Around a little bit. So, you want to get a copy of our talks too?
A little bit more about the topic of nutrients and switch away from upwelling specifically. But Rafe had a great idea actually that one place we might go do this is some places really already screwed up, like Pamlico Sound. So with that, that's all yours, Hans.
And we still have great seafood too, so you know that's. If you're willing to not worry about the contaminants. No, actually, it's very good. So I don't even think I'm going to mention the word upwelling, so please excuse me for not including the word. But hopefully, this talk will have some relevance with regard to where are we at in terms of the knowledge of nutrient phytoplankton growth, community structure interactions in estuary and coastal systems, and that's basically what I'm going to talk about. So if I don't finish it, that's what it's all about. All right. What do I need to emphasize that hasn't been emphasized already? I just wanted to point out that the marginal environments, which include upwelling areas, but also estuaries and coastal zones, account for about 70% of the primary production. So they are key environments. And that production is very much dominated by phytoplankton. And I just thought I'd show you that localized productivity on the East Coast with the two largest estuaries in the country, Chesapeake Bay and Alamo Pamlico Sound. And of course the reason for that is that they're nutrient enriched. And also, in the case, for example, of Pamlico Sound, some of these systems are hydrologically very good for high production. They're lagoonal, they have long residence time, so the phytoplankton can chew through the nutrients, and they're good places to look at what do you need to optimize production, what runs out first, and what happens when we enhance these systems with nutrients? What other kinds of issues come up? And I think that is relevant to the topics that we're talking about. So what do we know, more or less? And I'm going to emphasize a couple of these themes, but we're all very aware of the fact that nitrogen is obviously the key nutrient in terms of sustaining production in many of these systems, but there are a lot of systems that are N and P co-limited, and that's changing because of the way we're putting nutrients in or climatic effects that are changing nutrient limitations. So I'm going to talk a bit about that. And the other issue is that when we put a lot of nutrients in, particularly nitrogen and or phosphorus, you get other deficiencies. I know Ken will talk about iron, but silica is certainly one. And I'm going to give you an example of the Mississippi plume in the Gulf of Mexico that has led actually to phosphorus limitation in some areas. There's strong interactions between physical features in these systems and what the nutrients do, and I'm going to try to emphasize that. And these last two I'm probably not going to have time to talk about, but we know, of course, that excess production can, while it leads to more phytoplankton, can also lead to adverse effects in the environment. And if we have time for discussion, I can show you some things in terms of the linkage to hypoxia, but I think I'm going to stay away from that. All right, this is the famous Nixon regression between the input of inorganic nitrogen and primary production. Pretty convincing that nitrogen is the name of the game in many of our estuarine ecosystems. And I'm going to show you several slides now that involve actual experimental determinations of nutrient limitation using a variety of bioassays, mesocosms, etc. These are short-term experiments, usually no more than about a week or so at a time. These are ones that we did several years ago in two locations of the North Carolina coast, including right off Morehead City here, the inshore station, which is right in there, and then one out into the Gulf Stream itself coming by. And you can see that nitrogen does stimulate primary production relative to the controls. This is based on carbon fixation. Phosphorus doesn't do much. In fact, most of the time it doesn't do anything because there's plenty of phosphorus out there as phosphate. And then we did some work with iron. I'll talk a bit about that later. But, well, one interesting thing is that you often get different responses to different flavors of nitrogen. This is the nitrate versus ammonium added both at 10 micromolar, and you can see that often there is a stronger response to ammonium in these systems, and I'll talk a bit about that in the estuaries. And not much of a difference between nitrogen and iron, except if you use nitrate, as we do here, and add iron, you can see you get an additive effect. So there does seem to be a close linkage between nitrogen and iron limitation when you use a source of nitrogen that requires iron to be used. Whereas with ammonium, there is no significant difference. So you can see some of these nutrient interactions show up. I picked another system, which is the largest estuary in the world, the Baltic Sea. And this is work that actually we did with the Finns, and Pia Moisander published several papers on it. This is in the middle of the Baltic off the Finnish coast. And you can see here nitrogen is also the stimulatory nutrient, but here you get an N and P effect that's greater than nitrogen. You're going to hear more about that in other places. This is with regard to productivity, C14 uptake. This is with regard to biomass, chlorophyll A. More or less the same results. And again, no independent phosphorus effect. So nitrogen is generally the most important, but N and P comes very close behind. So, you know, the ratios are incredibly important there, obviously. Chesapeake Bay, this is data from Tom Fisher and his folks up there, where they look at nutrient limitation on a seasonal basis. And he has this very nice graph where you can see the different limitations that occur in this system along the axis of the bay, going from the ocean side out in here near the inlet up to the headwaters here, and you can see that in wintertime, when light is poor, light tends to be the most limiting nutrient. Then you get a period of high runoff coming down here from the Susquehanna and other rivers coming in. That leads to a lot of nitrogen coming in, and you get an N and P effect, and sometimes a pretty big phosphorus effect if you get a lot of runoff coming in. And that is basically a product of the high nitrogen loading that's occurring in the system. Then in the summertime, when the flow is down, much of the system is nitrogen limited, and then you go back again to the light and co-limitation and phosphorus limitation. So it's not that simple, and these nutrients do interact quite a bit and give an additive effect. Same is true in the Neuse River estuary where I work, part of the Pamlico Sound system. This is mesohaline waters that is held within the Outer Banks. So this is a fairly long residence time system, the Neuse River estuary. Nitrogen effect, in this case again, ammonium higher than nitrate. We use urea to look at DON as a surrogate. You can see there's an organic nitrogen effect there too. No independent phosphorus effect, but N and P tend to be a little bit higher than N. And if you're up in the creeks in the fresher part of the system, where we do get a lot of nitrate loading coming from the landscape, you can see there is a nitrogen effect, but the N and P effect is greater. So we don't want to ignore the phosphorus part of this story, I think. Okay, nutrient inputs have obviously accelerated greatly in estuarine and coastal systems. This is just kind of a potpourri of different systems that I've thrown in. Many of you are familiar with the Mississippi loading story, where both N and P have increased dramatically, but N has increased more dramatically, largely due to fertilizer use and untreated wastewater, but other places as well, including atmospheric inputs of nutrients in the coastal zone. This is from China, and then we have the Chesapeake Bay situation. So there, it's obvious that man is impacting the loading and ultimately the response of the system in terms of which nutrients are limited. So I thought I'd go to the biggest, to the mother of all watersheds and receiving waters, and that would be the Mississippi watershed in the Gulf of Mexico, receiving waters. And this system obviously has a very strong seasonal signal. When the rainfall is high, in the springtime, there's a tremendous influx of fresh water into the coastal system here. And you can see the dilution of salinity in here off the off the mouth of the Mississippi. When that happens, the N to P ratios change very dramatically, both total as well as dissolved, and this just gives you some idea of how dominant this nitrogen load is becoming from the land into the coastal environment. Those ratios are correct. That's over 1,000 for the N to P ratio. And this, of course, is coming largely from anthropogenic inputs. In the springtime is when there's the big bloom off the Mississippi plume region in the Gulf of Mexico. This is the N to P ratio that's occurring. So here you can see this big, huge freshet delivering all that nitrogen, giving very high N to P ratios. And then the system goes basically to red field or below red field even in the summertime. This is the algal production. And what you can see is there's this upswing early in the spring, but then after, actually before the N to P ratio decreases, you can see the production of algae actually decreases. And what I want to point out is that that is because phosphorus limitation has been induced in this system from the high nitrogen load. Okay, so here we're trying to stimulate something with high nitrogen, but actually you get to a point where you're not going to get any more because the system is becoming P limited. Not many marine scientists will argue that kind of thing, but this is just one of those, anthropogenically impacted places. So this is the results of bioassays that have been conducted by Jim Ammerman and his folks and others. And what that shows you is that in May, when that big freshet hits the Gulf of Mexico and you got very high N to P ratios and you do these bioassays to ask the algae which nutrients are limiting, well, the only stimulation they can get is if P is included in the treatments. Okay, so here the productivity is being held up, largely because there is so much anthropogenic nitrogen that's come into the system. Okay, so this is kind of the, well, it's the flip side of eutrophication here. If you want to increase things more, you have to start messing around with P. Now, if you're a water quality manager, what this is telling you, you got to reduce both N and P to really get the system under control. But I thought it's a rather interesting phenomena there. So I already told this story basically. Excessive anthropogenic nitrogen loading has led to P limitation, so that now the spring bloom is actually controlled by P supplies, which is kind of interesting. The management option in terms of controlling eutrophication is to obviously reduce N, but P as well probably, if you really want to control that spring bloom. And that's just, again, largely a product of how humans have changed these coastal systems. All right, I'm going to spend the rest of the time talking about nitrogen. And what I wanted to get into a little bit is the sort of flavors of nitrogen question. We know that nitrate obviously is the dominant anthropogenic input of nitrogen in these waters, but more and more we're having to deal with organic nitrogen as well as ammonium coming from all sorts of sources that are directly delivering these nitrogen sources, including animal operations, fertilizer applications, things like that. So it does make a difference, and I'll show you just a slide or two to show you what happens when we use different forms of nitrogen in the same concentration and then look at the phytoplankton community response. And the way we do this is to use diagnostic photopigments as indicators of the different algal groups. Okay? And that way we can get class level kinds of responses out of it. So if we do this experiment in the, this is Bogue Sound, so this is true marine water off the coast of North Carolina, nearshore water, and we add ammonium or nitrate at 10 micromolar, and then we measured the response using these diagnostic photopigments. So this would be the control, no nitrogen addition. This is nitrate and ammonium. You can see by and large there is preference for ammonium, but it varies quite a bit among different groups. So if you look at the total phytoplankton community, so this would be chlorophyll A, you can see there is a response. It's barely significant. Alizanthan for the cryptophytes, which are good food sources, by the way, There is a difference as well. Diatoms, somewhat of a difference, but if you look at the blue-greens, or the cyanos, they really have a very strong preference for ammonium over nitrate. So what this is telling us is that maybe from a management perspective, we really do need to look at this ammonium loading question, because it may very well be linked to changes in the structure of the community and ultimately food web changes. And this is one of the aspects of nutrient loading in coastal systems that people are looking at pretty intensively. The interaction of physical factors with the nutrient loads is another important determinant of who wins out and how the phytoplankton community is structured. So I've already talked about this part, but the other part I want to talk about is the hydrologic forcing that's going on in the system. And I think to some extent this is relevant to some of the upwelling questions and actual physical forcing of waters through in and out of these structures, because residence time is incredibly important in terms of determining who wins out when you have a nutrient loading or excess nutrient loading problem. The two things I want to talk about is residence time, that is the water residence time that it takes for the water to move, water entering the system to go out of the system. And imagine this being Pamlico Sound or Florida Bay, for example, which has a fairly long residence time. And so we calculate this in terms of water replacement. And when you have high discharge conditions with short residence time, the water is obviously moving through rapidly. That means if you're a phytoplankton, if you have fast growth rates, you win out. If you have a dry year, where nutrient regeneration is the dominant source of nutrients coming in, but longer residence conditions, other phytoplankton win out. And I just wanted to show you what this looks like. First of all, this is total chlorophyll A in the Pamlico Sound, lagoonal system, about year residence time. The arrow is the freshwater input to the system. So this is low freshwater input, this would be moderate, this would be high. In fact, this was after a tropical storm event, putting a lot of fresh water into the system. And this is going back to moderate. And what you can see is that the chlorophyll, or the phytoplankton biomass, is very much concentrated in the headwaters of these estuarine systems coming into the sound. But if you increase flow, you're adding more nutrients, but you're also displacing the population or the community. And then if you've had very high flow, the residence time is too short in these tributaries for the phytoplankton growth rate to really keep up with the flushing rate. So it's all happening out here in Pamlico Sound instead of in the sub-estuaries. And then here we go back to moderate flow. And if you look at that in terms of the diagnostic pigments that encode for several phytoplankton groups, actually we do about 5 or 6 of them. But I thought I'd throw greens in here, chlorophytes, which are fast growers. They like to grow rapidly when there's high nutrient loads, and so they can deal with high flushing rates versus some of the dinos, for example, and cyanobacteria. This is the hydrograph of the Neuse river water coming into the estuary. So this would essentially be discharge rate. And you can see when there's high discharge, some of the phytoplankton groups do well. For example, in a hurricane year like 96 or 99, where we had 3 hurricanes in a row, the green algae do very well, but the dinos don't do very well at all because they can't keep up with the flushing rate. So there's a direct nutrient effect, but then there's also a physical effect. And these two are interacting to determine what the structure of the community is going to be. Again, very important from the food web perspective. Chesapeake Bay, this is Larry Harding's work. Larry's worked with us on both the Pamlico Sound and Chesapeake Bay. And what I'm showing you here is the comparison of a dry year versus a wet year. So 95 was a relatively dry year. Spring runoff was quite low, so most of the production is really oriented towards these little tributaries as opposed to the main estuary. In other words, the phytoplankton are able suck out the nutrients early on in the process of the nutrients moving down the bay. Residence time is long, and then here you have the flood year, or the very high spring runoff year, 96, and you can see that most of the production is actually occurring down in the lower part of the estuary. High nutrients, and phytoplankton can take advantage of that, but they're also being flushed out at the same time. So if you look at that in terms of the composition of the phytoplankton, okay, here we have the dry year, spring and summer. Here's the wet year, spring and summer. You can see in the dry year, the composition is pretty diverse because pretty much everybody has a shot at the nutrients coming in. But in the wet year, it's mainly the fast-growing guys, the diatoms, that can take advantage of it. So just the flow regime itself has a very important structuring role in terms of the phytoplankton community. Turns out the nutrient loads were not all that different between these 2 years, but they were just at different parts of the estuary, and the flushing rates were different.
But Hans, it was, okay, the nutrient load was pretty much the same, but how much was the concentration in situ? Because the dilution was also a dilution in terms of the nutrients. So in some years you may see much higher concentration of phytoplankton may experience higher.
Yeah, but mostly up in the upper part of the estuary when the fast-growing guys are growing there. The nutrients are pretty low.
Always?
Well, they're moderate to low. So it's mainly who can grow the fastest to remove the nutrients, basically, in this system. And the diatoms tend to win out in fast, you know, under high flow, short residence time conditions. And then in the summer, when the flow slows down, you can see it sort of starts to come back a bit more to the lower flow conditions.
Hans, is there increased mixing associated with the, or is it more, any other physics?
Well, the mixed layer is probably depressed a bit more under the high flow, but only in the upper part of the estuary. You know, in these estuaries, when you get high flow, freshwater flow, the stratification sometimes is actually more intense. The salinity stratification actually can be more intense. So it isn't just wind mixing, it's also the salinity stratification due to the layering of the fresh water on the salt water. It's pretty complex. So going back to North Carolina, I thought I'd show you what diatoms do versus the cyanobacteria. And the cyanobacteria that we have in the lower estuary and in the sound are not the really bad toxic bloom-forming guys. They're mostly picoplankton and the small cyanos. And again, this is flow or discharge. So this is just 6 years worth of discharge data. And you can see that, you know, we have a lot of discharge differences due to storms and droughts. But that also makes for an interesting data set to look at who wins out under different flow conditions. And I'm using two pigments here, fucoxanthin, which is for diatoms, and zeaxanthin for cyanobacteria. So what I'm going to show you is the diatom blooms that occurred in the system. And you can see they occurred during very high flow periods. Okay. And then the rest of the time you can see the cyano blooms or high cyano biomass situation, and that occurred during the lower flow periods in the system. So there's a complex interaction between flow as a forcing feature and who responds and who can take advantage of the nutrient inputs to these systems. I think this is probably not unique to the Neuse or North Carolina estuaries. It's just that we have a good data set to explore this, but I think it's incredibly important in terms of structuring the phytoplankton community in lots of other places as well. And this is the result of if man gets into the act. So let me go back here. This would be in the periods, of course, where you'd have the very low flow and have the cyanobacterial dominance. And in Florida, they do such a good job of enhancing this in their lagoons off of the estuaries that they create high residence times that exceed the natural high residence times, and the cyanobacteria completely take over. So this is just a physical factor kind of situation going on. All right, the last thing I wanted to finish up with was really, I guess, most germane to this group, and that is based on this knowledge and other knowledge. You know, when we talk about enhancing marine fertility, there are two different sort of viewpoints here in terms of the nitrogen question. How do you get more nitrogen into these systems, particularly through the process of nitrogen fixation? Now, there's general agreement that N and P are the key nutrients controlling primary production. The geochemical argument is that you really only need to manipulate phosphorus, because if you have enough phosphorus, the nitrogen fixers can come in and fix the nitrogen that's required to meet the sort of stoichiometric ratios that you need in order to meet the production that you want out of the system. So the assumption here is that nitrogen fixation will fix enough nitrogen to meet your requirements if you have enough phosphorus to sustain the process. The biological argument is that nitrogen fixation is controlled not only by ratios, but by other factors like light, organic matter, iron, possibly trace metals, turbulence, grazing. In other words, it's much more complex than just having a ratio set kind of formula out there to get optimal production based on key inputs to the system. And this, of course, is the bottleneck, nitrogen fixation coming in to supplant the nitrogen needs in many of these coastal, but even open ocean systems as well. So recently, the limnologists have sort of gotten into this act by basically taking this sort of geochemical assumption that all you really need to do is control phosphorus, in this case to control eutrophication, because the system will fix enough nitrogen to meet the phosphorus requirements. Okay? And Dave Schindler and his colleagues, I think everybody knows the Schindler group and the Lake 227 group, published this paper back in 2008 that basically says, don't worry about nitrogen, just control phosphorus and everything will be cool. And then they put a question in the paper, or they posed a question of themselves and answered it themselves, and that is that, are these findings applicable to estuarine and coastal waters? And in the paper it's implied that it is, and it's based on this assumption that nitrogen fixation will supply the ecosystem with enough nitrogen to meet the nitrogen requirements. The assumption has been challenged, and I've been involved in some of these papers, but the most convincing paper, I think, to challenge this assumption is the one by Scott and McCarthy, and I'm going to show you some data from that. What they did was they took the data from Schindler's Lake 227 experiment. This is a 35-year data set where they manipulated Lake 227, experimental lake, with N and P additions, and then looked at the system response in terms of fertilization. What I want to point out is that they actually fertilized with both N and P in this part of the lake from 1968 through 1989, and then they only fertilized with P beyond 1989, with the idea that nitrogen fixation would keep the eutrophication level up and keep the production level up. So this data has been re-examined by Scott and McCarthy, and I'll just walk you through these data sets. So this line in here is when they changed course here. So they fertilized with N and P together, and you can see everything was going up all right. You know, total N, total P. The N to P ratio, you know, remained reasonably constant through that period. And then they fertilized with only P, based on the assumption that the system would fix enough N to keep the productivity going. Okay? And Scott and McCarthy looked at this data in a much more detailed way, and what they could see is that when the P fertilization continued, the total N actually dropped in the system. Okay? And the N to P ratios dropped in the system. And actually the total P concentration remained pretty much the same. So it turns out that there isn't enough nitrogen being fixed in this lake to keep up with the eutrophication that was induced by only P additions, okay? Interesting part of this story, you know, when people start actually looking at this data in a very detailed way. This kind of experiment, I think, really needs to be done in the marine environment in places as well. Now, we don't have 35-year data sets and long-time fertilization, but I think it's a really good test of the fact that N fixation does not meet ecosystem requirements when you're into fertilizing systems to try to up the sort of trophic state. It's not because there aren't enough nitrogen fixers around, and this will look very familiar to John. This is just a NIFH tree showing you all the different taxa that have the nitrogen fixing gene. They're everywhere, and there's lots of them around. But the thing that needs to be taken into consideration, is, I think, is the biochemistry and physiology of nitrogen fixation. Now, if you just, you know, going to make the argument on N to P ratios, and if you put enough P in, you'll fix enough N, and, you know, this looks pretty good. But the problem with this process is that there's several constraints that go beyond just the N to P ratio to control it. And one of them is energy requirements, either by light or heterotrophy, which is the one up here. The other one is other limiting factors, particularly iron and maybe other metals. And then you have this issue with oxygen and turbulence in the system that may create suboptimal conditions for some organisms to be able to fix nitrogen. So it ain't that simple. It's not just a matter of shoveling in enough P to get the fertilization done to get your nitrogen requirements to optimize your production. And this is the system that probably gets the closest to that, and that's the Baltic Sea, where they have huge nitrogen-fixing blooms during summertime. This is a mix of, these are all, well, it's a mix of nitrogen fixers and also some non-fixers, but the dominant nitrogen fixers in here are Nodularia, Anabina, and Aphanosomna. And what folks there have found is that at best you can only get about 30% of the nitrogen you would need to meet ecosystem demands just by nitrogen fixation. So these systems do depend, well, this is again the flip side of the eutrophication argument. You know, if you want to up the production more, adding more nitrogen is going to help. Okay? And this is one reason why we want to reduce nitrogen inputs to many of these systems, even if they have nitrogen fixers in them. And the other factor I wanted to point out is one, again, from one of Pia's papers, is that turbulence is incredibly important. This is a graph of the mixing depth, the upper mixed layer in the Baltic. This is when they had Nodularia and Anabina blooms, and this is the rate of nitrogen fixation that was measured with different mixing depths. And you can see that it drops off as the mixing depth increases. So as the organisms are mixed down into less favorable waters, the ability of them to keep up the nitrogen requirements for the system slows down. Now, there are some other factors at play there too, but that's the key one, is that turbulence matters in here as well. And the final proof of the pudding that you do need external nitrogen inputs to these systems to get as much production out of them is just the flux, are the budget estimates. And this is data from a paper I published with Thad Scott in ES&T a couple of years ago. Again, this is taking Schindler's et al.'s data in Lake 227, rate of N-fix, rate of denitrification. The net N-flux is out instead of in. So Lake 227 could use more nitrogen, folks. We want to get the production up. So this indicates that N-fixation will not do the trick just by itself. You need external nitrogen inputs. And then there was some data that we worked on from Lake Mendota, Okeechobee, Lake Erkin. And then if we go to the marine environment, and this is really data from lots of different people's papers, including Doug Capone's and Wolf et al. In the Baltic, and Mike Peeler, you can see that the percent of nitrogen needs that you would be required that would have to be met by N-fixation is pretty low. The system that gets the highest or closest is in the Baltic, where you have the most optimal conditions for these blooms to form. You have that strong summer stratification, stable conditions. But it's only for 6 weeks of the year, you know, or so. In some years, it's not even that. So the end fixers, you know, work hard and they try hard during that time, but still you need the external nitrogen inputs. And the last thing I wanted to show is, I don't know if anyone else is going to show a trichodesmium slide, so I threw one in there. Of course, is the open ocean nitrogen fixer, trichodesmium. Now, John and all have shown there are other nitrogen fixers out there. But trichodesmium definitely has a requirement for iron when it's out there, and you're lucky enough to do an experiment with it. Here you can see this is chlorophyll, and this is acetylene reduction, an indicator of nitrogen fixation. This is with iron additions to, this is Gulf Stream water off the coast of North Carolina during a trichodesmium bloom. You can see an iron effect, no molybdenum effect, and this is iron and molybdenum together. It's basically an iron effect. So there are these other requirements out there that could have an impact on enhancing marine nitrogen fixation potentially. Those are largely coming from atmospheric sources of iron input by dust, for example. So, just to sum up, I think we're still pretty much in agreement that nitrogen is the name of the game, but because of human impacts and over-fertilization of nitrogen, some systems are showing P co-limitation, or they're even P limited. So the N to P ratios are important, but they're also very dependent on loading, external loading inputs. Excess loading of one nutrient can cause another one to become limiting. We've known that from lab experiments for quite a long time, but we now have field data to show that. And there's this strong interaction of physical forcing features and nutrient enrichment that determines not only the total production, but who is actually responding in the structure of the base of the food web. And lastly, I think I've pointed this out now, both in freshwater and marine systems. We don't think that end fixation can meet the requirements for optimizing production. And that's controlled not only by N to P ratios, but other factors, physical features of the system, and mixing depths. So I think I'll stop there, and hopefully I've made up a little bit of time, and open up the floor for any questions or discussion.
Hans, I think one of the things that you're pointing out, which is really important for this issue, is primary production of the wrong species. It doesn't count for a fish, right? And so I guess one of the questions is in upwelling areas, Francisco.
Actually, that's that slide there.
Yeah, I mean, may know is sort of how predictable in upwelling areas is it that you're going to get the right kind of algae? Is that something that just happens all the time, or do you risk getting algal species that aren't the best for fish?
I'm not sure how well we know the answer to that. We typically always get, initial Margulis Mandela, but 95% of the time you get diatoms. Now, there's centrics and pennates, and they vary in their, and occasionally we get an unusual condition where there's a lot of nutrients and very little mixing, and red tides can form. Now, again, I don't know, in the fishery literature maybe they have more information, but the early fishery literature information suggested that actually dinoflagellates were not necessarily bad things. That the first feeding of a fish larvae requires a certain bite of certain quality. We tend, in coastal upland systems, we tend to get diatoms as the primary organisms, and the same has been true with most of the iron fertilization experiments. The concern has been raised with those that in most cases pinnates are the dominants, and there's some concern that those organisms may produce toxins that would not necessarily be toxic to the fish, but could be toxic to in other ways. If you look in the open ocean, you tend to find just mostly pinnates in very low value, so they're the ones that are hanging around. While the centrics tend to be hanging around on the coast.
You think that's a product of also like fast, you know, just being able to grow fast and being able to take advantage of it? Yeah. So it's very similar to this kind of, yeah.
I mean, you basically the same thing.
But do you think that oceanic systems and oceanic coastal upwelling systems are much more consistent with respect to what's going to bloom like diatoms than, say, estuaries, where you could get all kinds of things blooming depending upon the situation? Although sometimes they are predictable, like San Francisco Bay, you can predict when a diatom will take place there.
It's so different from this place that Russ was talking about.
But my take is that the estuaries tend to have a much more varied group of organisms that bloom
And
the coastal systems.
not quite as well understood? Or
Well, I think it's complicated. I think the physics are very complicating in these kinds of systems. Jim would be able to show you the flip side for San Francisco Bay. You know.
Hans, you talked about physical factors and nutrient forms as drivers of changing phytoplankton community composition. What about interactions between species? Selective grazing, interactions between owl cats and allelopathy, those kinds of things. You didn't say anything about those processes.
No, but they are important, particularly when we get flagellates, for example, and dinos that can, you know, be both autotrophs and heterotrophs at the same time. And we have seen some of those interactions, so I think they are important. And you're right, I didn't talk about them, but I think they are. They need to be put into the mix too.
One more comment. I like this slide, but in a way, it's correct for, say, an estuary or for many of the systems, but Francisco Chavez showed earlier this morning that off of Peru, you've got hypoxia, suboxic conditions right below it that's sort of concentrating the zooplankton, concentrating all the fisheries. And so in that case off Peru, where you have hypoxia and oxia with one red fish, off of Peru, you would have 20 fish
Right.
under those same conditions of hypoxia.
Yeah. Now this is much more laid out for a shallow
Yes.
water system that has strong bottom water
With
stratification.
benthic fish and benthic
Right.
feeders and crabs and everything else.
Right, and where they use the habitat basically.
Right,
Much
right.
more.
But off of Peru, there would be, 20 or 30 fish under the hypoxia box.
One of the things that Hans mentioned to some extent, I missed the last part of Francisco, but is the stability of the systems. The hypoxia and the gradients are very stable, so you have an ecosystem that can evolve in that perspective. And in estuary, I would expect the system to be much more dynamic and not allow that system.
Yeah, well that's one of the major perturbations that we see actually in terms of big switches in blooms is the either onset of extremely stable conditions for after an event that's occurred, like a major storm or a flushing event. And usually it's after, it's when that happens that you get a unique bloom of something, you know? Like the good example is Carlidinium, this toxic dino that we don't like to have. It loves blooming after a storm that hits, say, in the early fall, but not too late in the fall. so that you have the strong salinity stratification that sets itself up, and you still have enough nutrients and you still have enough light for it to be able to bloom. So timing is everything when it comes to those kinds of events.
Hans, is there commercial fishing in the Pamlico system?
Yeah, the Pamlico Sound is big crab, benthic crab fishing. It's actually most of the crabs that people eat in Maryland that are so-called Maryland crab cakes, they actually come from Pamlico Sound. And shrimp, flounder, bottom fishing. The system is very intensively fished.
So do you have any idea where this place fits on the Scott Nixon plot of fisheries harvest versus primary production?
It's pretty high. The other thing is that Pamlico operates very well on regenerated nutrients, you know, during the during the low flow summer period, because it's lagoonal and the residence time is long. So, you know, it just is able to go through a molecule of nitrogen much more efficiently than many other systems. So that's what makes it such a good habitat, you know, a nursery habitat, I think. But it is intensively fished. Now, there are some fish that only have their juvenile stages there and then they're caught offshore, you know, like the mackerels and the more coastal fish.
I'm thinking of a comment that you made, Francisco, and that is, I remember an ancient paper by Reuben Lasker, who talked about the fact when fish were recruiting some of the, I forgot whether there was sardines or anchovies, there was like a week or two period. If they had dinos just the right size, you got a big year class, otherwise you didn't. So the timing is everything and the species is everything.
Actually, it goes that way with menhaden. By the way, in the news, you like menhaden there. It's a big nursery for menhaden, but it's very timing oriented. You can have really bad years or good years for menhaden depending on the sequence of these events that I've talked about. But there's enough menhaden around so that I guess they, you know, can keep up with climatic adversity.
Andy, your little diagram that said that the dinoflagellates like it warmer and slower.
The cyanos.
Yeah.
Yeah.
Right. And I've seen the situation in upwelling where the whole upwelling system was controlled by dinoflagellates, and it seems like if you get relatively slow upwelling, and it's not enough to get the diatoms going, dinoflagellates can actually control the whole thing by vertically migrating. We've seen this off Baja, California.
Yeah, that's neat.
And you could tell the time of day looking over the side of the ship, and it's red, and it must be noon.
Yeah.
But they basically would be sweeping the nutrients out of the water column every time they went up and down. And they produced a canopy of light. This is dark underneath, actually, so diatoms had no chance for that. So there kind of could be a sequence in upwelling rates that determine what the species composition would be during a particular time.
I kind of imagine an upwelling system as being an estuary turned on its side. You know, this way instead of that way, where the residence time is incredibly important in terms of the circulation of the nutrients in the system, because you got the light field then too, at the same time.
Just a little question for Lisa. For your project, getting back to that, does the sustainability or timing of fish production for oils and stuff like that, does that make a big difference? Do you need to get, you know, do you need to harvest just once per year or every month, or does that make a difference?
It does, although it's so early days at this point that we don't have a really useful opinion. But fish farmers, for example, have a problem, which is they get burps in activity that make it very hard on their labor. This is not all bad. Like a lot of farmers in the wintertime, they kind of enjoy sloughing off a little, and their workers get to take a winter break. But In a perfect world, you want your people and your equipment used continuously with kind of minimal up and down time except for some maintenance. So in a perfect world, you'd be harvesting every day.
Which is an interesting question, just to bring back the odd timing of how this talking about others. You know, what's the window here, you know?
But fish farmers we've dealt with, that like the first site that you saw, they were looking for things for their workers to do in the wintertime because they didn't want to keep them if they had nothing to do, but they didn't want to lay them off, and what would you do?
To what extent do you think the community is grazing controlled and not nutrient rate controlled?
Well, we're actually looking at that question now, but I think that it can be very important in the lower estuary where it's operating on regenerated nutrients, as opposed to the upper part of the system where all the fast growers and the diatoms often can dominate. In the lower estuary, grazing controls, I think, are really important in the summertime, very important. And everything is small because the nutrients, the lower Neuse and that region, the lower Neuse going into Pamlico Sound, that's sort of where everything's been taken out because of the long residence time. It almost looks like an oligotrophic system sometimes. It's all picoplankton and very small diatoms. And the grazing in there, Mike Wetz, who actually did a postdoc with me, he found phenomenal grazing rates in there. So I think I think it is important.
Although you make the point, not in the regions of high, fast-growing diatoms. So in the models that we work with, we have both fast growth rate for diatoms and there's a small advantage that they have from a grazing point of view in terms of the silica crustule. And we find that the former is the more important in ensuring diatom growth on the coast as opposed to some other phytoplankton in the model. So I think it's consistent with what you're describing. In the fast-growing regions, it seems to be more bottom-up driven.
Yeah.
But there could be some selection, perhaps.
Yeah, there's a seasonality issue there too, because the picos, which are very much dominated by cyanobacteria, they really peak out in the summer. Their only real act in this whole play is during the summertime when it's warm enough, and they can have fairly good growth rates, and they're being grazed like crazy at the same time. So the system is just really operating very, very speedy at that time.
Following up, I think it's pretty critical to consider the food web setup and dynamic. And we've been talking a lot about translating the phototrophic biomass in fish, and there is evidence of isoslime, jellyfish in Chesapeake Bay and Gulf of Mexico and many other places where this doesn't necessarily goes in the food chain always to fish, but sometimes the carbon is differently utilized in a way that is not then harvested in for any good use.
Yeah. Yeah, this is like a very super conceptual kind of thing.
Yeah, no, but
we actually get blooms of jellies in Pamlico Sound, at certain times of the year that graze down things very effectively, where the fish don't, the menhaden are out of luck at that time, because they're being outcompeted by jellies that are actually doing grazing at that time. Again, that's dependent again on residence time, because if you have high flushing rates, well then the menhaden do just fine.
As far as I can tell, the world has more than enough jellies at this point.
So I propose we go take part in our own food web. and try and be back here about 1 o'clock. Some of the thoughts that are being cast around are, not only to perturb the nutrient dynamics, but to somehow be able to seed or control the phytoplankton response so you don't get unwanted things, and so you do feel the food chain. So maybe you can talk about that while you graze.
We can barely dominate anything in Charity's 96 well plates, so we have no illusions about us dominating anything in a larger scale.
Yeah, but if you are able to see your system with the population,
That's our.
then you know.
Francisco, how frequent are jellyfish
Blooms in the.
Jellyfish are very common on all the upwelling areas. They tend to, I mean, you know, leather bacterials come all the way from Indonesia to eat jellies in Monterey Bay. And the Australian ones come to Peru. So it's a common component of the ecosystem.
Do you think it's been increasing or it's always been?
They're always there. I think Haddock's data from here suggests that there might be a slight increase. But I don't know that off Peru at least they've had a systematic. One thing is you think they're increasing because you see them more often, right? I mean, how much of this is real and how much of it is you're looking more? Yeah. But they are very common components, and they have been implicated in I don't know that they directly feed on fish larvae, but they're certainly not a good thing for fish larvae.